welcome Professor Vasta. Dr. Vasta was a pioneer in many aspects during his career. Really, he introduced to our field of marine diseases, among other things he does, some of the new notions derived or even leading in other species research, model species, I'm thinking in higher vertebrate mammals research. So he brought into our field a fresh look at some of the host-pathogen interaction. His research was a great source of inspiration for many people in our field, including myself. I still remember some of the first papers published by Professor Vasta in the 70s. It's been a long admiration, I think, on my end. Anyway, we are very delighted to have him around, joining us for this speech. Professor Vasta has two doctoral degrees, one in zoology, one in biochemistry. That gives you a little information on the breadth of his interests in science. From University of La Plata in Argentina, he came to New York State actually for a postdoc up in Buffalo for a couple of years before joining Musk Medical University of South Carolina for his first position before moving to Baltimore for the last 13, 14 years, 20 years, where he now works at the School of Medicine of the University of Maryland. You will see a little change in the title. Dr. Vasta thought to focus his talk today on recognition of self and non-self by organisms.
Well, thank you very much, Bassem. And again, I want to first thank Bassem and Emmanuel and all the colleagues, that invited me to be here. It's a great pleasure and an honor. It's my first time at Stony Brook, and given the reputation of this institution, it's a real pleasure to be here. And again, I should apologize for the change in the title, but to some extent, I did it on purpose in terms of trying to incorporate, in addition to oysters, other organisms that I felt would have a wider sort of interest for diverse audience here. So again, I hope that you will not disagree with the move. Anyway, my lab is interested in mechanisms of molecular recognition in terms of microbe consortial interactions, in particular pathogens or parasites and their hosts. And particularly on the carbohydrates, that the role that carbohydrates play on this. Can you hear me? Can you hear me? Okay. And you may wonder why carbohydrates should be interesting. We have heard about antibodies, we have heard all kinds of recognition mechanisms, but why sugars? Well, because if you look at the cell surface, this that looks like a poorly maintained lawn, let's say, it's actually a cell surface. And this stuff that you see here is the glycocalyx. This is the way a cell looks from the outside. So you have all this fuzzy stuff here that are sugars, basically oligosaccharides coming from glycoproteins or glycolipids that are on the surface. So these oligosaccharides encode very complex information. If you combine, if you do permutations of the different sugars you can have in these oligosaccharides, you have more variants than even proteins. So there's complex information, and this is encoded by carbohydrate-binding proteins, which by doing so modulate cell-cell or cell-extracellular matrix interaction that, again, has a consequence in function, in early development, the self-recognition, or innate immunity in terms of non-self-recognition. So if we go into innate immunity first, one of the, again, innate immunity, if you're not familiar, is a type of immunity that's very fast. It does not require antigen processing. Like when you get vaccinated, it needs a couple of months to develop a good immune response, whereas we could all be dead by then, unless we had innate immunity, which we have, fortunately, and this is very quick, in minutes, 2 hours. But one of the striking aspects is the variety of receptors that mediate recognition, or innate immune recognition, versus adaptive immunity, and by this I mean immunity as mediated by antibodies and B cells and T cells. That's typical of the vertebrates, so to speak. That is very slow again. But you know, you have immunoglobulins and T cell receptors, where in innate immunity is a very large number of receptors that are involved in a good variety of cell types, instead of B and T cells only. And you have soluble lectins, for example. You have mannose binding receptors, and DC sign and lectin-1. All these are membrane lectins. You have NK cell and scavenger, complement, and toll-like receptors on the surface. These are TLRs, together with a NOD, for example, signal via NF-kappa B to activate immune genes, for example. So each one of these families is very diverse in itself. So for example, if you talk about TLRs, it's between 12 and 15, depending on mouse or human. So there's a tremendous diversity of receptors that mediate innate immunity. In case, as you can imagine from the title of this talk, I'm going to talk about lectins, which are the sugar-binding proteins, and then can mediate not only host colonization by pathogens, like pili and adhesins, you know, from bacteria and so on, but also pathogen recognition by the host, commensal interactions in consortia, symbiosis, and then we'll see at the end of this talk subversion of pathogen recognition by these protein-carbohydrate interactions as well. So then what is a lectin? It's a question for those of you who are not in this field. The lectin is a protein that's characterized by a carbohydrate recognition domain, or CRD. So it's a protein that recognizes sugars, basically. That's a lectin by definition, from the Latin legare, to bind. And they can also have other domains which are structurally and functionally diverse. So you have basically more bang for the buck with a single protein. You have multiple functions in the same polypeptide. And they can be membrane associated, usually with transmembrane domain, or humoral, where you have, for example, they're soluble, and this would be the cartoon of a mannose binding lectin, for example. This is a very important lectin in humans. They have a collagenous tail and cysteine-rich domain. Now, lectins have been classified in multiple ways of using different factors, like, for example, calcium requirements. C-type lectins require calcium, most of them. Galactins do not, and or their specificity for different sugars. But since the early 80s, by Kurt Rickemer, who identified the first sequence motif to define a lectin family, again, we have a C-type sequence motif, S-type, P-type, etc. So more recently now, we can classify lectins and identify them and characterize them by the structural folds. Every structural fold is unique to a particular lectin family. Here we have the pentraxins, like C-reactive proteins, C-type lectins I mentioned before, P-type, galactins, I-type, etc. So now, not only a sequence motif, but really a structural fold can define the lectin family. Now, as I said before, these are the C-type lectin fold, and as I said before, they are chimeric molecules or mosaic molecules by combination with other domains, recognition or functional domains. And the C-type lectin family is, I think, the prototype example for that, because you have here the C-type lectin domain is the red hexagon, you see? Whereas all the other domains that you can see here enable, let's say, classification of these lectins in multiple groups, multiple subfamilies, but at the same time, they underscore the functional diversity of these proteins. A single protein can accomplish multiple functions. So that's important to keep in mind. So how does the C-type lectin work, for example? How do these lectins recognize sugars? Well, if this is the mannose binding lectin, this is a typical, what you call a pattern recognition molecule, and that's the concept of Peter Janeway and Ruslan and Medvedov, you know, by which innate immune mechanisms that are based in pattern recognition recognize structures or patterns that are absent in the host. And that's why it's so quick. You don't need to adapt anything. We naturally have proteins that will recognize anything that is not in our cells. Basically, if you have a pathogen, a bacterium, a virus, a fungus, they will have surface characteristics that are recognized immediately by these proteins. So these are pattern recognition molecules. And the reason is because, for example, the MBL, mannose binding lectin, recognizes 2 equatorial hydroxyls in 2 neighboring carbons. That is the pattern that's recognized by the mannose binding lectin. Mm-hmm. So this could be in a sugar, for example, in mannose, this is carbons 3 and 4. Mm-hmm. In a oligosaccharide chain, for example, in the microbial cell wall, you have now these the oligosaccharide chains with mannose at the end that are recognized by trimers of this lectin, forms trimers and then trimers of trimers that can recognize microbial sugars that are separated 45 angstrom apart. I mean, different lectins will recognize different patterns. But the advantage of this interaction is that these interactions are relatively low affinity. But the oligomerization of these proteins produces high avidity. So they bind very tightly, even if they are low affinity. Okay, the different then domains that you see in some of these proteins can function by, for example, tagging a particular pathogen. In this case, a bacterium, for example, would bind to mannose residues and then bind to the surface of a macrophage. Basically, it's what's called the opsonic effect. Basically enhances phagocytosis by recognizing the surface of the macrophage. Now, other domains, for example, in the collagenous tail, will bind to this serine protease, mannose binding lectin associated serine protease, long name with a MAS acronym. This will trigger activation of C2, C4, and then C3 of the complement cascade. The complement cascade, again, it's an innate immune pathway that leads to either further opsonization with some of the complement components bound to the cell surface, or the lytic pathway where recruitment of other complement components leads to a hole in the bacterial wall. So basically you have, and it's interesting because we consider the classical pathway by the mammalian immunologists, the ones that's triggered by antibody antigen, C1q and C2 and C4. But you have complement and sea urchins, and corals and tunicates, you have complement components. So the classical pathway should be the lectin pathway, actually, or at the most the alternative pathway, because in reality, the complement pathway was basically hijacked by the adaptive immune system, as we went through the development of the vertebrate lineages. So if there's something classical here, it's really the lectin pathway. So still there's many questions that these proteins that I've just described as binding a very fixed pattern. And in fact, I must say that, for example, with what I said about the mannose binding lectin, you're going to find 2 equatorial hydroxyls in 2 neighboring carbons in other sugars, like fucose and mannose. So really, the mannose binding lectin is a misnomer. The mannose binding lectin binds equally well fucose and glucnac and so on. So that has to be kept in mind. But it's like Boyd wrote in the early 50s, and I'm not that old, I was very little. But the paper was called The Specificity of the Non-Specific. And the problem of specificity of these lectins is not that lectins are non-specific. They're exquisitely specific because they can recognize 2 orientations, or the orientation of 2 hydroxyls. If one is axial and one equatorial, it doesn't bind. But the problem is that that particular pattern, molecular pattern that's recognized, is present in other sort of molecules, organic molecules. Okay, so if the recognition of specificity is so fixed, and for many lectins it's truly hardwired in the germline, how does this work for immunity? Because you expect this tremendous diversity that we have kind of imprinted in our minds when we think about antibodies. We have tremendous ability to So if this is the case, but the fact is that invertebrates which lack antibodies and B cells and T cells have been equally or more successful than us in terms of surviving infectious disease. So antibodies are not the answer, and innate immune mechanisms on which they rely for fighting infectious disease are very efficient. So if this is the case, how is diversity in self and non-self achieved? Because there's multiple bacteria, there's multiple viruses, there's parasites, they're mutating, they're changing, and most of you are familiar how rapidly these pathogens change. How extensive are the lectin repertoires in a given species, and if they have evolved in the functional diversity. If their targets are evolving, are these proteins evolving as well? Mm-hmm. So my lab has tried to address some of these questions using a variety of models. I mean, mostly non-conventional models, although we use mouse for influenza and other studies on infectious disease. But I would like to illustrate some of these questions with a lectin one. A student in my lab, Eric Odom, identified, which gave rise to a whole new family of lectins. And this is a striped bass. You must be familiar with this, or rockfish, Moronius saxatilis. And he identified a lectin, which we call FBB 32 kilodaltons. It's 32 kilodalton protein, binds fucose, and expressed in the liver, released to plasma, inducible by infectious challenge. So it works as an acute phase protein, like a C-reactive protein. When you get an infection, in less than 2 hours, you release from your liver a lectin, which is called a C-reactive protein, that binds to anything, basically, but the levels in plasma increase about 2,000-fold in 2 hours. That's why it's used in pregnant women when they break waters, for example, to figure out if there's an infection or not. That's the, it has been used for the last 100 years and keeps being used as the most sensitive method to find out if there is infection or not. Because if there is, boom, the CRP levels will be very high. In any case, so it functions like this. It's released very quickly to the plasma. What was interesting, when he sequenced this, he couldn't find any similarity with any of the known lectin families I showed before. It's a long story, but to make it short, he identified 2 lectin domains, which we called F-type domains because they bind fucose. And this is the situation that we saw in the Moronius saxatilis, in the striped bass, and it's also present in the zebrafish, which is a good model for biological research. So this is a dual CRD. You have 2 CRDs in tandem. But there is one single CRD in tachylectin from Tachypleus tridentatus. That's basically the Japanese horseshoe crab. And the eel. And this is a very interesting protein because thanks to the eel lectin, Morgan and Watkins in the early 50s also discovered that the blood groups, the human blood groups of the ABO, are encoded by sugars. Fucose for blood group A, 0, basically, galnac for blood group A, and galactose for B. So, the discoidin, this is a protein that is from the slime model, Dictyostelium discoidium, a very good model for development and cell adhesion. Xenopus tropicalis with 3, and the trout on Coryncus mycis with 4, and the long pentraxin from Xenopus laevis with 5 in the pentraxin domain. The Chrysostogyrgis bindin, which is a fertilization protein, a gamete recognition protein, and Drosophila, the ferrite-like receptor. It was published incomplete at Johns Hopkins because it had been published up to here with a C-lectin domain, 2, 3 or more sushi-like domains, but this was missing. So when from the genome, we were able to confirm that our sequence was correct with an F-type domain and more sushi domains, complement control domains. Actually, this was also published incomplete, the one from Xenopus, and this is the one that was identified. So you can see that these proteins are interesting because not only they have multiple F-type domains tandemly arrayed, but also they are chimeric proteins. They have other domains, like in discoidin. I'll talk about it later. So since this was a new family of lectins, we were interested in the structure, of course, and in collaboration with Mario Bianchet and Mario Amsel at Johns Hopkins at the biophysics department, we crystallized the eel lectin. And here you see the subunit where you can see that this is a typical beta barrel, with 2 beta sheets, one here with 5 beta strands, antiparallel beta strands. In the back, you can see the other beta sheet with 3 strands, you see antiparallel strands. Then you have a short sort of beta sheet forming the bottom, and then on this side you have a few what called alpha 10s, short alpha helices that kind of bind the cation. We'll talk about it later. And here you have all these loops that bind the fucose. So not only this was a new lectin family, but it's a new structural fold for the lectin family. So it's a new, basically for the animal lectins. I mean, that was pretty interesting. So here you can see that more clearly, how you have one beta sheet here with 5 and the other one in the back. And then you have these loops, that go back and forth between the beta sheets, forming the binding cleft, which in this sort of a potential representation, you can see here, the loops, and here the fucose in this positively charged cleft. And each subunit binds a fucose that you can see here, and the physiological species is a trimer that's held with a chlorine here, stabilized by arginines and so on. So this was quite interesting, and you may wonder, well, which residues bind the fucose? And I don't want to bore you with a lot of structural biology, but it is very interesting that they bind this axial hydroxyl. The residue is histidine 52. You have arginine 86 and arginine 79 in almost a perfect tetrahedral configuration here. Then there's a very rare disulfide bond here formed by 2 neighboring cysteines, 82 and 83, that form a van der Waals interaction with carbons 1 and 2. And then there's another interaction with the oxygen of the ring, etc. But they're very, very well defined polar and apolar interactions between these lengths. And so the only reason why I'm showing this is because I want you to get a good sense of how specific are these proteins. And each residue interacts with a particular hydroxyl in a particular orientation, axial or equatorial. So that's the basis of the specificity. Yeah, you have the coordination of the calcium. Again, it's bipyramidal, pentagonal, and in the case of this calcium, so we're not going to get too much into that. But of course, fucose is not floating around, in the environment. Fucose is bound to oligosaccharides, many times as a terminal sugar. In the case, for example, of Lewis trisaccharides or H trisaccharides, remember that I told you that this lectin binds blood group antigens. Well, here you see now an oligosaccharide. You have the fucose that I showed with the 3 residues that are key to bind this axial hydroxyl. And here you have the H substance. So you see that in both, the fucose is bound by the same 3 residues, and you have the disulfide bond here. But it's interesting that the specificity for the trisaccharide then is now established by this loop, what we call the CDR1, which actually interacts via these 2 residues with the 3rd unit. The same with the tyrosine here that interacts with the hydrogen bond. In the H substance, these 2 are reversed. This gal is subterminal to the fucose, and glucnac, which is here, is now the 3rd residue. So the interactions are very similar. It doesn't bind other Lewis. The only reason, for example, Lewis X, because in that case, this acetylamide group, from the glucnac, is pointed in this direction. And this loop is the most rigid of all. So it's just the steric hindrance that does not allow the recognition of Lewis X versus Lewis A, even if it's recognizing the fucose and the galactose and the glucnac. So the very specific and sometimes very small details that we'll see later are key to this. Anyway, we moved into the second in complexity, which is a 2-domain, and this is a more saxatilis in terms of the structural aspect. We crystallized it, and the structure was resolved again at very, very good resolution. And you can see now you have 2 CRDs linked with a linker peptide. The physiological species is again a trimer. If you see it from the top, it's almost identical to what you saw in the eel. But you have these connecting or linker peptides here. So we have a cylinder now, 81 angstrom by 23 angstrom, with 2 faces where you see the fucose bound. Now this is quite interesting because now you have a protein that can cross-link sugars on one side and other sugars on the other side. What is mostly interesting is that when you compare the 2 binding sites of these proteins, the NCRD or N-terminus CRD, carbohydrate recognition domain, is much more similar to the one I described before, the eel. Here you see the disulfide bond, with the fucose here. You see the 3 residues that form this perfect tetrahedron, binding to the axial sugar. In this case, for example, there's no disulfide bond. Now we have phenylalanine to 20 which is much more bulky, you know, and tilting the sugar out of the binding cleft. And you had this phenylalanine. This it's a mistake. This should be phenylalanine 37 actually, and this is replaced now by this tryptophan, which is also much more bulky and kind of closes the binding site. So in other words, not only, although the binding site for fucose remains, the extended binding site to accommodate oligosaccharides is different. And here you have the N-terminus ERD and the C-terminus ERD with the, compared to the eel, to the one I showed first. So basically what I'm trying to say here is the two faces of the cylinder are different. And the reason is very unusual in proteins that instead of organizing in the oligomers as head to tail, they're organized head to head and tail to tail. So you have one face of the cylinder with 3 binding sites with different specificity as the other face of the binding site. You can see this in this representation where you see that this phenylalanine 168 and glue 167 form this much wider ridge, which is connected with the glue 184 via the tryptophan 183. So this narrows and closes. These are like level curves, you know, from geology. If you cut, here they are, you see. You cut, this is from the side, so you have different planes where you're cutting the levels. And you can see there's ridges and valleys, like you would see in geology. And here you have where the fucose sits, and here you have the disulfide bond. That's kind of a flat apolar surface. And here you have phenylalanine 220, very polar, you know, and much more protruding structure. Anyway, this basically is to illustrate how these ridges and connections kind of limit the binding of the extended binding site. So here you have a comparison now of the eel agglutinin, which form dimers, but linked by non-covalent interactions, compared to the monosaxatilis lectin, which is also a trimer, but linked here, where you have 2 different faces, and here you have the same, basically. It's the same subunit. This is reflected here. And we have, I mean, the information we have obtained is that they bind microbial ligands, not only L-fucose, also L-galactose, but the 2-acetamido-fucose, the colitose, very common in bacteria, and rhamnose as well. Well, they also, as I showed, the host ligands bind H and Lewis oligosaccharides. So basically, now you have a protein that can cross-link host glycoproteins on the surface and microbial sugars in this model, where you will have, you know, different from the MBL. You would have now a CRD of one type and a CRD of another type binding to bacteria and then binding to the cell surface. And this experimentally can be done in the past peritoneal macrophages, where if you pre-incubate with the recombinant protein, you get a much higher phagocytosis rate. If you include fucose in the medium, you bring it down to the control. And this is the PBS, no sugar bound. And it goes to a dose response, meaning the more you add, the higher increase in phagocytosis, and then it goes for a plateau. So I've talked about these 2 arrangements, the eel-agglutinin and the striped-bass lectin. And you remember that Xenopus tropicalis in the trout you have with 3 and 4 CRBs. So these are models of how these lectins could be arranged, which would confer a tremendous capacity and recognition. When you think about all these different planes of sugar binding surfaces that you would have. Another level of diversity in recognition is given by the fact that each one of these lectins have isoforms, have isolectins. So in the eel, there's at least 7 with point replacements in the sequence. So the highest sequence variability, I'll show this in a minute, is localized to the loops that encircle the sugar binding site. And the amino acid substitutions really reveal the binding specificity. It's not going to dwell too much into this, but for example, these are the 7 isoforms of the eel-agglutinin, and E1 and E7, for example, replace in this loop that you remember binds to the 3rd sugar of the oligosaccharide, replace all these, or most of these residues by smaller residues, less polar. So now, instead of being rigid, as I mentioned, that could only recognize Lewis A and not Lewis X, now it's flexible. So that gives a much broader specificity. The same with the other sugars in CDR2 that form a hydrophobic pocket that binds to C6. I mean, so what I'm saying here is basically the different isoforms now will have different, basically, sequences, and the lighter the color, the more variable the loop. Meaning that most replacements are in these loops that recognize the fucose, as you can see here. So each isolectin has a different fine specificity for different sugars. So we don't have the diversity you would have with antibodies, but with the different arrangements of subunits that I showed before and the isoforms, you can generate tremendous diversity. Finally, to bust this myth of that lectins are really hardwired in the germline and there's no recombination, basically that diversity is not generated by genetic mechanisms. You have the binding. Now this is a gamete recognition protein identified in Zebrafish Drgall-L2, which ended up being a member of the F-Lectin family. It was not identified as a lectin until very recently. And here you show, and we can see that the structural diversity here is by positive selection that happens in this same region. Here you have the overlap of binding without the G. This binding is the name of this protein. Here you have the Agglutinin that is superimposed beautifully. And again, all the variability takes place here by positive selection. You have recombination at several introns, but you also have alternative splicing. So each individual will have transcripts that show 1 CRD, 2 CRDs, 3 CRDs, 4 and 5 CRDs in tandem. So you can see that a single individual can have transcripts of all these. Interestingly enough, not all these are made into protein. So this is a single individual at transcript level, but this is at the protein level. And you can see that a single individual will make 1 or 2 or 3 at the most. Of course, the sequences are different in this area. And it's proposed that this protein, it's meant to inhibit polyspermia. During fertilization, each male can basically recognize and cross-link ligands on the surface of particular eggs, otherwise it can't fertilize. And once they cross-link, they block the entry of other sperm. So that's how you have this population dynamics. The other combination that is quite interesting in this family is this scoidin. Okay. And this is an interesting protein, again, that was initially postulated as a lectin in a PNAS paper, then came another PNAS paper saying it's not involved in sugar plant, whatever. It's a very, very conflicted protein, but eventually it is a very interesting one because once we crystallize this, we realize that the scoidin forms a trimer identical to the one we found in the striped bass. Now I'm talking about the slime mold, and you see the similarity in structure between these two. And here you can see that the scoidin is actually composed with an F-type domain. Here you see the two trimers, virtually identical. And then the other domain is not an F-type domain, but is an H-type domain. This is the lectin from the Helix pomatia. This is the escargot, you know, this is the edible snail. In the albumin gland, they have a lectin, which is, what was called by Uhlenbruck a protectin, because when the eggs are laid, the actual cell is coated by this secretion from the albumin gland, which is, I would say, 90% lectin, and it was called protectin because it was postulated that this would protect the eggs from infection. Because the lays, again, the eggs of snails, as you know, they're laid on the ground. So anyway, very interesting because now we have in a slime mold a lectin that forms a trimer with one domain, when one face is an F-type domain, the other one is an H-type domain. So this talks about evolution and exon shuffling through evolution as well. So we have now, I gave examples, what I'm talking to summarize is you have Lectins of the F-type that can be two phases that are identical, like in the E-lectin, and again promote opsonization, or two different phases with a linker peptide in the striped-bass lectin and promote opsonization. In terms of fertilization, from 1 to 5 CRDs that can cross-link then glycans during fertilization. And in cell adhesion, now you would have an example in the dictyostelium of a lectin that binds the cells to its own secretions, the mucus. And you know, when the amoeba in dictyostelium migrates on its own mucus, so that would be the function of this lectin. So, to summarize this, the structural and functional diversity of these repertoires as an example of most lectins is given by the diversity generated by tandemly arrayed FCRDs with different carbohydrate specificity, multiple isoforms, including recombination, alternative splicing, other mechanisms with distinct binding properties. The oligomeric quaternary structure with variable binding avidity. Basically, you can generate different avidity. CRDs from different lectin families. F and C, for example, like in Drosophila ferret, you remember a C-type lectin domain and an F-type lectin domain. H and F, like in the dictyostelium, and then pentraxin as well in the Xenopus long pentraxin, where you have F-type domains and pentraxin domains. And of course, effector domains that are the ones that do the job. And if lectins may have diversified to mediate different biological roles, like opsonization, microbial pathogen, fertilization, cell adhesion, etc. So I still have time, I believe. And I would like to move now to a different lectin family, which are the galectins. I hope you're not totally bored or asleep at this point. I'll try to go fast on this, but it's interesting because these proteins are tremendously conserved in evolution. Very, very conserved. You find galectins in fungi, you find galectins in invertebrates. We have plenty of galectins, and I'll show some examples that are so key to our immune function that it's striking. But this is the first galectin structure that was published, and we did it in collaboration with Osnat Herzberg at the same institute. And this was from a mammalian galectin. And this shows a dimer of two subunits held by a non-covalent bonds. I mean, this is a hydrophobic region, has been crystallized with N-acetylactosamine. This is a disaccharide, which is a galactose and then N-acetylglucosamine. And they come in three types. Prototype, which is this one, two subunits, again held by a non-covalent interaction. The chimera type, where you have one single CRD and an N-terminus tail very rich in proline and so on. And these can form oligomers, can make trimers or pentamers, based on binding through the tail. And then you have the tandem repeat, which you have two CRDs bound through a linker peptide. A little bit like I showed before for the striped-bass lectin, where you have two CRDs, globular domains, bound with a linker peptide. And here you can see, well, and look, LACNAC bound, and here you can see more detail. This is a cartoon that explains it all from that original paper, basically, where you have certain hydroxyls recognized by certain, this is dying, certain residues directly through polar interactions, or sometimes through a water molecule, like in this case, where they interact with the N-acetyl group of the subterminal sugar, which is N-acetylglucosamine. I point that out because it's quite interesting. These proteins are made in the cytoplasm, and they can be translocated to the nucleus, where they can join the ribonuclear proteins and form part of the spliceosome, basically, in terms of the protein synthesis. They can also be secreted to the environment through an unconventional mechanism. They have no leader sequences. And there they can interact with the extracellular matrix. You know, galectins bind galactose or lactose or LACNAC. So they have a common specificity, and they can bind in cis, you know, binding its own sort of sugars on the surface, extracellular matrix. And this led to the idea that these are developmentally regulated proteins. I mean, they participate in development. We work in the zebrafish, and this is just to show that they really do We characterize the repertoire of the lectins in the zebrafish. As you know, the zebrafish is a very important model for biomedical research and for any kind of research, basically, because it's multiple advantages. The embryo, you can work with the embryos 2 hours after fertilization, or 30 minutes, basically. Everything is transparent. You tag the genes. You can see the gene products, all over the embryos. I mean, it's very spectacular. And of course, it has several prototype lectins. One deletion isoform as well that we either purified or we clone. One chimer, actually, there are 2 now, and there's like 3 tandem repeats. So the 3 types are represented in the zebrafish. What we found is that one in particular, 2, the L2 form of lectin 1, is only expressed in the notochord very early. Here you have the actual probe, here you have the sense probe, the control, here you can see higher magnification. And here you have the in situ, this is instead of hybridization, this is immunostaining with an antibody against that protein. Here the validation of the antibody, the crude extract and the purified protein. And you can see how the lectin localizes between the notochord cells. Now the notochord, as you know, is a central engine of development for the zebrafish and for us. I mean, it's no different. From the notochord, you get all the signals, basically, that will lead to development of floor plate, of neural tube, of the myotome, via hedgehog signals and wind and all kinds of, so for bone development, for circulation and so on. So it really is leads to everything, basically. So it was interesting that this localizes here. And of course, what we did was what you do with zebrafish. You inject the embryos with antisense oligonucleotides to knock down the gene. And the first thing that we saw is that with the mismatch, the control, nothing happens. It's perfectly normal. With the antisense, if we block that one, we get a myotome that's completely disorganized. So this is a higher magnification of this. You can see the myofibers that are beautifully organized. Here you can see the nucleus. This is the red muscle, the size, skeletal muscle. And you can see how the fibers are disorganized now. So definitely it's involved in muscle development. Here is the co-injection with the cRNA to rescue the phenotype. So basically what you do is you knock down the gene. It's not a total knockout, but then you co-inject with the RNA to rescue the phenotype and show that this is specific effect. The surprise came when we looked at the heart. In the same knockdown, this is the mismatch, which is obviously the control. We have the ventricle and the atrium forming here in the embryo. When we look at the knockdown, you only see the ventricle. There's no atrium that's formed. And then when you rescue the phenotype, you get again the atrium and the ventricle. So it's not only involved in skeletal muscle development, but also in heart development. Collaboration with Peter Hitchcock, Madison, we also looked at retinal regeneration, to show not only development, but tissue regeneration. And in the retina, the Müller glia, you have stem cells there. If you damage the retina with light, basically what you see here, you have the expression in the morpholino knockdown. There's no expression of the galectin that you see in the mismatch. Actin control. You can see that the recovery of the Müller glia is much faster in the mismatch. And here you can see with antibodies against the DPC, this is basically an antibody specific for the cones, regeneration of the cones. Here you see the mismatch and the morpholino, no difference. Does not affect the cones, but with an antibody against rhodopsin, you see a reduction in the rods. So basically what's happening here is that when you have retinal damage, the Müller glia, these stem cells, secrete galectin-1. This is done in the zebrafish, I forgot to say. So it not only works in development, but also in regeneration, because the galectin is secreted, and you knock down the gene, you don't get rod regeneration with the same speed as you would get it in the wild type. So definitely they are involved in development, but also they are involved in immune regulation. And this is a work by Gabriel Rabinovich in Buenos Aires also, who found out that they can also form lattices, cross-linking glycans at the surface. And this is one of the most striking examples of galectins being involved in Th1, Th2 polarization. because depending on the activity of a salil transferase, basically, as I said, lectins bind galactose. And if they basically are masked by sialic acid placed there with the salil transferase, you don't have galectin binding and you don't have any proapoptotic signals. If you don't have expression of salil transferase, the galectin binds and kills these cell populations. So you get Th1, Th polarization. So that was a striking paper. More recently, we have found that also these galectins, instead of binding only self, everything that I described was binding to self-ligands. Development, retinal regeneration, immune regulation. But they can also bind glycans on the surface of pathogens. And this, we have found, we know now they bind to the GP120 of HIV. They bind to a bunch of LPSs, you know, like meningococcal and gonococcal and so on, and Haemophilus. They also bind to the type 14 polysaccharide of Streptococcus pneumonia, bind to Leishmania. This is the LPG, the lipophosphoglycan, the LPG of Trichomonas as well, and Schistosoma mansoni as well, the LDN, like the NAC, and the oligomannans of Candida. So they can bind fungi, viruses, bacteria, and so on. So we have a protein that not only binds self, but also binds non-self. And that is a little bit of a paradox, but I would like to finish this talk. So anyway, we can place now then galectins in terms of these innate immune receptors. And I would like to go now into the subversion of pathogen recognition via the galectins. And this is, again, to bring oysters into the play now. Some of you will wake up and we have typical before and after pictures, you know, it's like these commercials for diets. You know? Here we have a mesocosm where you have introduced some of these microalgae, like Isochrysis and Tetracelnis, and oysters will clean up the water in minutes, you know, and you all know that they're key components of the estuarine waters and maintain water quality and so on. But what do the oysters do with these macroalgae? Well, it's food. They eat them and they process them all over. Whatever they can digest extracellularly in the gut, they will digest intracellularly by phagocytosis by these hemocytes. The problem is that together with the algae, sometimes they also eat Perkinsus, this parasite that has wiped out basically the oyster populations, both native, wildlife, and farm populations all along the Atlantic coast and the Gulf Coast. And here you have the trophozoite and the zoospore with the apical complex here and the insect. I mean, when you talk to some Plasmodium people, they will say, but this is an apicomplexan, you know, this is, because, you know, it's so similar to Toxoplasma or to Plasmodium anyway. But I'm not going to dwell into this, but early on, with a student in my lab, we found that, you know, the hemocyte can, when we were able to culture these parasites like other labs, you know, culture them in vitro. And then you could do in vitro exposures of oyster phagocytic cells, equivalent to mammalian macrophages, to the parasite. And it's a bit counterintuitive because you would imagine that the parasite tries to escape, you know, avoid recognition, but it seems that they eat this more than anything else, you know, and they eat so many. Here you can see 1, 2, 3, 4, 5, 6 in the process of being eaten and so on, to the point that they will detach from the substrate and float there. The hemocyte is full of the parasite. It's very interesting. Anyway, the question for us was what makes this parasite so palatable? You know, what is the recognition mechanism that takes place there? And again, I must say, clarify here, that's not the only one. There's multiple mechanisms. But one that's related to what everything I discussed before is a galectin, like the one I mentioned in humans and the cow and in the zebrafish. In the oyster, we have a galectin now with 4 CRDs in tandem. What we saw in the mammals, you know, is up to 2. Interestingly enough, these are extremely conserved. Here you see the overlap of the bovine, you know, the cow galectin in gray, with the 4 CRDs of the oyster, 1, 2, 3, 4, overlap the binding sites. Here you see the tryptophan 68, the histidine 45. So all the residues match perfectly. So there's perfect conservation between the oyster and the cow, and humans as well. I mean, the only difference is this tetrapeptide here that's houses these two residues that I mentioned before that were key for interaction with the N-acetyl group of the N-acetylglucosamine. So this whole tetrapeptide, you can see this whole loop is missing. You know, here you have the 4 CRDs, and this is a cow. So these residues are missing here. So this lectin has broader specificity, basically, because now the N-acetyl group is not a determinant, basically, of specificity. In any case, without going too much into this, the interesting aspect is that this broader specificity can be reflected in the binding to environmental bacteria. Now you have again a lectin that perhaps it binds self, but interestingly enough, it binds a tremendous variety of non-self, meaning environmental bacteria of all types, and with different intensities. The taller the bar, the stronger the binding of the recognition. It also binds the algae, isochrysis and tetracelmis, particularly tetracelmis, isochrysis very poorly recognized. But what binds the best, the strongest, are all the different strains of the parasite. These are all Perkinsus strains and species. So it's very interesting that this protein that binds the food and binds the pathogens, you have to remember that obviously oysters do not have immunoglobulin, so they recognize pathogens via lectins. And the binding is specific. Here we have the binding to the trophozoites with bioimmunofluorescence. And here this is inhibited with digalactoside. This is a disaccharide for which this galectin has very strong affinity. This is with triple sugar, doesn't do anything, and this is just the protein, without the protein. So the binding is via the carbohydrate recognition domain. And where are the transcripts? Well, the strongest signal we got was from the hemocytes. Again, these cells that are macrophage-like, they will eat everything. And that for the oysters are both defense cells as well as feeding cells. So we have them in the gills and the heart and the muscle and the mantle, but all at a similar level, which suggests that all these tissues are soaked basically in hemolymph. So it's most likely that the signal comes from hemocytes that are present there. Well, we isolated hemocytes, and all the signal is in the cells, but in the plasma, there's nothing. This is in the circulating hemocytes that have been collected in wet ice. So when the hemocytes are moving around, they have a rounded shape, and there's no galectin in the plasma. Now, when we let them attach to plastic, like put the blood in a dish, now we see that there's still the signal in the cells, but now the galectin has been released to the environment. So that was quite interesting. So they decided to look by immunofluorescence, and here you see the rounded hemocytes that are circulating. Here with DAPI, here is the Alexa, and they need to be permeabilized. So without saponin, you don't see any signal, meaning that the protein is inside the cells. You need to put a detergent basically to let the antibody go in and bind it. So that's why you don't see anything here. And these are the controls and so on. Now when the cells attach to plastic, they take this shape, like a typical macrophage. You can see phylopods, these very thin pseudopods. Now again, these are permeabilized with Triton X, and you see the stain in the cytoplasm, very strong. But the difference is that if you don't permeabilize them, you see all the stain at the surface of the cells. So not only the galectin has been released to the environment, but it bound to the cell surface in cis. And you see very clearly with a very high density of galectin in the phylopods. So this is a merge, with the nuclei, with the DAPI. So that was quite interesting, because now we have cells that are self-responsible for recognition and immunity in the oysters, expressing and releasing this galectin. And with an antibody against this galectin, we could reduce the phagocytosis rate. So not only it binds the parasite, it binds to the cell surface. And here I want to point out now that you have a lectin with 4 CRDs that we showed that it binds non-self. It binds to bacteria, it binds to the food, it binds to the parasites, but also binds to self. All these glycans on the surface of the hemocyte, of the phagocytic cell. So that's what the paradox I'm talking about, and you can inhibit again this phagocytosis, and the model we are proposing is there's multiple receptors, both for the food and the bacteria and so on, But the parasite, because it has this very high affinity for the galectin, it outcompetes anything else because it binds so strongly and uses this to enter the cells. Whereas other receptors perhaps can still function with other mechanisms. So this is a little bit of a model that we developed in the lab, that where in any surface where you have hemocytes, in this case is the gut lumen, for example, but hemocytes is like our macrophages. You look at the lung, and you're going to have lots of macrophages walking around there, just making sure that the bugs that come in through breathing, are going to be eaten and killed, So oyster are the same thing, and every surface is basically flooded with hemocytes. But these trophozoites that go in, to the gut, for example, would be taken by these 4-CRD galectins, and then these would be endocytosed. And I didn't talk about this, but we have identified all this basically antioxidative machinery of the parasite, superoxide dismutases. It doesn't have catalase, but it has ascorbate-dependent peroxidase. It has all the tremendous effective machinery through which it blocks the respiratory burst, with which the oysters and we kill every pathogen as soon as they get in. Well, the parasite has a fantastic antioxidative machinery with all these proteins. And that's a matter for another seminar, but the idea is that the parasite survives intracellular killing, migrates through the epithelium, and then goes in, into the host, or produces systemic infection, and eventually they kill the host. The reason why I highlighted the gut interactions here is because I wanted to make a parallel with Leishmania. As you know, Leishmania is a very important parasite, particularly in tropical regions, where the amastigotes are taken by the little sandfly, Phlebotomus paladasi, for example, for Leishmania donovani, and these are taken in the blood meal, but instead of being flushed through the gut and the rear end of the sandfly, they are captured by these galectins in the midgut of the insect. So this is a galectin produced by the fly that the parasite, now with all this galactose exposed, has taken advantage of. So they stick here and they mature now into the promastigotes and so on. They develop the flagellum, and while they do that, they start capping the galactose with arabinose that is not recognized by the galectin. So that releases the parasite from the midgut, and now it can swim with this nice flagellum to the salivary glands, which will be put into the new host once the sandfly bites. So it's a nice parallel how parasites are so smart that they can evolve their glycons, the glycocalyx, to match the host or the vector, in this case, to mature and take advantage of all that. In any case, just to finish, related self-recognition versus non-self-recognition. The Jane with Mechitov model is a pattern recognition of receptors recognize pathogens through microbial-associated molecular patterns. They used to be called PAMPs, but all microbes have these patterns. So it's not only the pathogens. Instead of pathogen-associated molecular patterns, now we call them MAMPs, which I still disagree with, but it's okay. Now these MAMPs, these patterns, are absent from the host. Now the paradox is galectins bind similar self and non-self patterns, and it's interesting because this is probably based on how galectins are compartmentalized and oligomerized and how ligand is presented. So it's not only galactose there, but as I showed before, it's a matter of to what oligosaccharide they are, attached. And I think in many ways it depends on a lot of sort of thermodynamic considerations in terms of how the water participates in the organization of the CRD and how hydrophobic is the surface, etc. I'm not going to go into that, but as an alternative to this, and because galectins are so conserved, as I mentioned, in evolution, what we proposed was more the Trojan horse model for some. So the idea is not that this model is not accurate, but like everything else, there are exceptions. And you have to be clear to make sure that you don't generalize, because then you lose the ability to discriminate these wonderful adaptations, in this case of the parasites, to their hosts, which is taking advantage of very conserved proteins, like the Lectins, and pathogens and parasites have evolved their glycones in sort of a parasitic mimicry, which is well known in bacteria, but it seems to be very well known in parasites. So, as I said, phagocytosis may sound counterintuitive, but in reality, the parasite has evolved its glycone to use these proteins to enter the host. Why? Because it has this fantastic antioxidative machinery through which it can block oxidative stress. So basically enters, can be killed by the host, and then lives happily ever after and proliferates. So, and to acknowledge here lab members, past and present, and collaborators, and including the people at Johns Hopkins, Rabinovich in Buenos Aires, at Herzberg, Mosena, Bassem, and Emmanuel here at Stony Brook, and others, and grant support from different agencies, and thank you very much for your patience.
Thank you, Gerardo. Any questions for our speaker?
Sorry, I can't hear you.
Don't people speak louder, I think.
Do you have any examples of maybe some of the hosts on the organs that are attacked by those parasites to produce anything to get rid of those collectors or inhibit the way of attachment to the host?
Well, as you know, I mean, this is an excellent question from a biological standpoint. And for evolutionary biologists, you could kind of put in the frame of the Red Queen effect, basically, which is, like the Red Queen tells Alice, in order to stay in the same place, you need to run as fast as you can, which is kind of a paradox in terms of coevolution within consortia. And in terms of microbes and hosts, there is this competitive evolution in terms of the glycans, for example. Apparently, like the hosts are trying to modify in evolution, the glycans or exposed sugars to prevent adhesion of pathogens or parasites through their pili, fimbria, adhesins, etc., through all these sugar recognition proteins from the pathogens. At the same time, the pathogens would be doing exactly the same to avoid being recognized by the hosts. And then you have these examples in which the pathogen does exactly the opposite, modifies the glycans to be recognized and to enter the host. It's very interesting because now there's a paper where HIV enters at the lymph node level via a lectin 1. And it's very interesting because, at the mucosa, it needs the Langerhans, so the dendritic cells. It infects dendritic cells, and then it's transferred to the T cells. But at the lymph node, which are very rich in galectin, the galectin binds to the GP120 and binds to the cell surface of the T cells, and then promotes longer stay of the virus on the cell surface, and eventually gets in to CCRX5 and so on. So the cell puts all the machinery in play now to get rid of the virus and to kill the virus unsuccessfully. Now it's quite interesting because this is a recent virus relatively, so it's a virus that, and it keeps mutating and so on and so forth. But you can see how quickly evolution takes place in how a parasite or a pathogen can adapt to the virus, because even in a T cell, which has all the machinery, to kill, it's not able to. And for example, we work with influenza, and we see a similar situation there at the lung epithelium. And you would not believe the cytokines and all the machinery that's triggered upon entrance to the epithelial cell. But it's not that the virus is smart. I mean, that's teleologically speaking. You're talking about selection, basically. Some particles will have pre-adapted, so to speak, to being vulnerable to all these. So it is certainly very interesting. The same way the parasites can release some of these glycans, like Plasmodium, that release all the surface molecules as, you know, like the octopus would do with the ink, you know, sort of, how do you think with an English, decoy, a decoy. You know, and to divert all the immune response to that, and then they switch, you know, antigens, and then you have a new parasite. So the same thing, you know, host and parasite throw everything at each other, you know, the kitchen sink, basically. And what those that survive are the ones that maintain this. Otherwise, we wouldn't have pathogens, unfortunately, you know, and we do have. Yeah.
The thing is that if you're a good parasite, you don't want to kill all your hosts.
Right, I agree with that, but this is something that is something that we humans have concluded in terms of what we rationalize as why a parasite is a parasite, or why a pathogen is a pathogen. But you know, I think you're right on that, and you know, some people analyze this in terms of the equilibrium, like the parasite and the host have not reached yet the equilibrium. But in the case of Perkinsus, for example, it's very interesting because the oysters live in reefs, you know, they are one oyster next to the other one. So in this case, and they filter feed. Basically what they do all the time is filter water and so on and so on. So when you look at the Perkinsus oyster interaction, it's not the same, and that's why I think the risk of generalizing some of these things. When one oyster dies, and all the tissues basically decompose, or they are basically teared apart by the scavengers and everything. So you have a lot of Perkinsus trophozoites floating in the environment, which are going to be filtered into all the neighboring oysters. So in fact, that the parasite kills the oyster promotes spreading of the parasite. And the other factor is that once exposed into the seawater, many of these species, or most of the Perkinsus species, will sporulate. They go into a phase which is a hypnospore and will divide. And you get hundreds of zoospores from a single cell. So now, you have to think that exposure to the seawater is an advantage to the parasite. So to some extent, you come to the conclusion to this particular parasite, it is an advantage to kill the oyster, to spread, to apply, so to speak, all the spreading and infection mechanism of the parasite. So in principle, I agree with you. And you can see in most parasites, you know, that perhaps just if I can take 2 seconds to give an example, which is extremely interesting, if, you know, for all biologists, but these are proposals that come to NIH, you know, and there's a lot of proposals coming in nowadays of purifying components from nematodes, from worms, you know, that, as you know, people that are infected with gut worms do not suffer from Crohn's disease or any of these autoimmune diseases. Why? Because these worms, again, have been very smart in evolution, you know, in developing, you know, immune inhibitors, immune suppressors. So there's a lot of therapy going on nowadays with people suffering from Crohn's, where they feed them with eggs of these worms. Well, when you're at NIH sitting in a panel there and you read this, it sounds pretty brutal, you know, and say, well, in this country, you know, that people are doing this, you know, to patients. It's particularly bizarre, but these people do very well. And I guess if you're suffering from Crohn's disease, you know, forget about if you're in an advanced technological country or not. You want to be cured. So many of these proposals are trying to figure out what are the worms doing to immunosuppress locally, because these people do not become like an immunodeficient patient, you know. So and this goes to your comment that the bugs are smart enough to suppress the local epithelial immune response, but not the systemic immune response. So these people survive and live happy, you know, with the worms and protected from these autoimmune diseases. And so this is an example that supports the claim, but I think it doesn't apply to all the examples.
One last question, and then we'll move to lunch to finish up the discussion.
Sure.
Yeah.
I think it's a follow-up to the same question. In symbiosis, as we know that sometimes plastids, for example, in plants, they came there because of symbiosis and mitochondria in, have lectins been shown to play a part in this, or?
I'm not sure what the question is. Both mitochondria and plastids are all products of secondary symbiosis, so that is well known. Whether the interaction between the host and the symbiont took place via lectin is very likely. I mean, now there's a lot of reports of lectins found in dinoflagellates, in these microalgae. And it's, and some of these dinoflagellates eat algae from which they incorporated their plastids. So it's a likely possibility, I would say. It's very difficult to prove that. But in corals, for example, which they are symbionts with, as you know, the microalgae, there's quite a bit of evidence that this interaction initially takes place via galactose-binding lectin. Then they go into the archenteron, in the gastric cells, and so on and so forth. But they apparently, and that's why they are so specific. Every coral species has a specific zooxanthellae, how these algae are called, to which they associate. It's not that just coral just filter in any algae. No, it's absolutely specific interactions. It's not just like that. So in summary, the reason for showing all this structural stuff that obviously some people may be interested, many others not, is just to illustrate how these interactions take place by very, very subtle molecular interactions. So that's why, to some extent, I disagree with this pattern recognition, because it gives a sense to those who are not in the field that it's just a pattern. It's something that's a shape, but it's not true. I mean, it's molecules. You have their molecules, and the interactions either polar or hydrophobic, or hydrogen bonds or whatever. So I think it is misleading to use it in this way, and that's why I'm concerned about it, because I've seen people that think, that lectins are completely non-specific, that they bind something that's, just has one particular shape when they bind to it, which is, again, lectins are more specific than antibodies in a sense, because they can recognize, just as I said, orientation. Not even the presence, just the orientation, axial or equatorial to two neighboring hydroxyls. That's all they recognize. So, anyway, that's.
Thank you. So lunch will follow, and of course, you're all welcome to join.